So a chi-squared test is a statistic that we use for this IB course. There's a second one we're going to use called the t-test that we'll cover later on. But in general, statistics are mechanisms which allow us to make unbiased decisions about whether to accept or reject a particular conjecture. And in science, we call conjectures hypotheses. So regularly in science, what we will do is we will do an experiment and we will collect data from that experiment to compare our data against an expected hypothesis, or sometimes even an expected theory that is out there. So what we then do with our data is we calculate a chi-squared value, and I'll show you how to do that within this video, and we compare our calculated chi-squared value from our experimental data against a so-called probability value, or p-value. Now, the p-values will allow us to assess whether or not the results that we've got, an association, is due to chance, or whether the results that we've got from our experiment actually support the hypothesis which is there. The chi-squared test can only be done with categorical data, and I'll explain what that means in a second. But what the chi-squared test allows us to do is look specifically at the degree of agreement between sample data and a so-called null hypothesis. And so we'll go through these steps here one at a time. But before we do that, let's have a look at this slide here. Here we've got the syllabus statement, and the syllabus statement is specifically asking you to look at using a chi-squared test to look at two species and whether or not the two species have got some kind of association between them. Is there a dependence between these two species? What the syllabus statement is also explicitly saying is that you're expected to apply the chi-squared test having done some quadrat sampling. And at IGCSE, you did quadrat sampling, so I'm hopeful that you remember all of that, but you might need to revert to the notes further back. So if two species are typically found in the same habitat, they show what's called a positive association, and that association could be a dependent association or a non-dependent association. So for example, if you've got predator-prey or a symbiotic relationship of some description, maybe a pollinating insect with the presence of a flower, then what we've got is a positive association between those two species which are living within that habitat in the same place at the same time. I also want us to think about a negative association, and we'll come back to the idea of a negative association when we look again at niches and we look at this idea of competitive conclusion and partitioning. But remember that the niche is the whole entire profession of an organism, where it lives, how it lives, what the interdependent relationship with other organisms are, what the interrelationship with the abiotic environment might be, etc., etc. And if two species occupy exactly the same niche, then the two species are going to compete to such an extent. And remember, we call the competition between two species is interspecific competition. If we've got two interspecific competitions between groups which have got exactly the same niche, one species will outcompete the other. And the so-called loser will have to migrate. Its numbers will fall if they don't migrate, and in some cases they will go perhaps to numbers which are too small and they could become extinct altogether. We call this idea competitive exclusion. But if organisms occupy a very similar niche, but there are slight differences, then what can happen is something called resource partitioning. And resource partitioning will allow the organisms to live together in the same environment and just live in what's called their realized niche as opposed to their fundamental niche. I'm going to ask one of you to explore this a little bit more and do a presentation like we've done so far. But what we're looking at with the chi-squared test are two species. They're found in the same place. Do they have an association? Are they interdependent? We're going to explore species by using quadrat sampling, and that is what this is all about. Quick recap on quadrat sampling then. What we're going to do is take our square or rectangular piece of material, and we're going to place that randomly, that is without bias, at a particular point, and we're going to count the number of organisms which are found within that square. There are a number of rules that we need to make for doing that, and we are going to repeat the sampling process. The more repeats we have, the more accurate or more representative the data will be, and hopefully this will allow us to see enough data and look for an association between two particular species. So this is coming from BioNinja. It shows you, well, the quadrat is square. The organisms that we count can't move too quickly. They're non-motile or sedentary organisms. We need to place the quadrat randomly. So what we'll do is grid the area, and we'll give coordinates along the horizontal and the vertical. And then what we'll do with a calculator is generate random number coordinates. So perhaps the random number coordinates might be 2, 2. So we go across 1, 2, we go down 1, 2, and that allows us to get that particular coordinate. Then we decide how we're going to place the quadrat with that 2, 2 coordinate. And what we're doing here is we're taking the bottom right-hand corner and placing it just so. Once it's placed down, we're going to count the number of species A and count the number of species B. If a species is on the edge of the quadrat, we make up rules. If they're more in than out, we count them in. If they're more out than in, we don't count them within our categories. So that's the idea. What we want to do here is we want to have quadrats which are placed randomly to avoid any kind of bias. So what you can see here is kind of zoned, clustered. You might see the species that you're looking for, and you might want to place it there. You're doing this perhaps not consciously, but by using random number coordinates, you are going to get a random sample, and a random sample will or should give you a representative sample. So let's go back to this starting slide. We need to think about these 5 steps. I'm going to take you through one at a time, but to remember that we're looking at categorical data. Categorical data are data sets which are in categories. So species A versus species B, and the frequency count is going to be the number of species A versus the number of species B. One thing that you need to appreciate, however, is that the chi-square test can be powerfully used in other circumstances, not just comparing the association of two species. So I've highlighted this in your textbook by using a slightly different example, because you need to be able to apply the chi-square test potentially for an internal assessment. So let's think about plants, and let's think about the height of plants and whether or not we've got our hypothesis that we would expect plants to be taller if there's more light. Because there's more light, there's more photosynthesis, and therefore more generation of glucose, which allows more respiration for ATP so that the plant can grow. Now, what we're doing here is we're looking at height. So we can't measure individual heights. What we need to do is create categories. So we've got tall plants and we've got short plants, and we need to have some kind of rule that allow us to distinguish between the two categories, and that will allow us to have categorical data for tall versus short. We can also then say, well, we're going to define what we mean by shade and non-shade, and we've again got categories that we can use to count our data. So again, categorical data is what we are using if we're doing a chi-square test. We're going to start off with a hypothesis, and there are 2 sorts of hypothesis. We've got a null hypothesis and an alternative hypothesis. So in science, we need to be open-minded. We don't need to be always looking for results which confirm already established theories. The idea in science is that we want to try and actually progress the scientific enterprise by being open-minded and looking for evidence that suggests that perhaps the theory that we've got is the wrong theory. So we've got 2 different hypotheses, the null hypothesis and the alternative hypothesis. Then what we've got to do is do our experiments. We're going to construct a table of frequencies, the number of observed organisms of species A versus species B, or the number of observed plants which are tall in the shade or short in the shade, etc., etc. And then we're going to look at something called the expected values, which I'll explain in a moment. Then what we're going to do is apply a formula, and that formula will give us what we're going to refer to as the calculated chi-squared value. We then work out something called the degrees of freedom, which won't make sense now, but will hopefully at the end of the video. And then using that degrees of freedom, we look up some probability tables for chi-squared, and we're going to look at a p-value and we're going to compare our p-value with our calculated value to establish whether or not our results are due to chance or whether our results support or reject the values that we've got there. And I'll explain this 0.05 in just a second. But these 5 steps need to be applied. And just to take away that we can only use categorical data for a chi-square test, and your specific syllabus statement is asking you to look at data arising from quadrat sampling. So let's look at part 1. So what we've got to do is have a look at quadrat sampling. So we've got the different quadrats which are placed down here, and we put into a table what we've got. So we're looking at the possible association between the queen scallop and the king scallop, and we're looking for whether when we put them in our quadrats, whether they're present or they're absent. And these are the frequencies that we find in the table, and we've got the total values here and the total values across. So we're doing random sampling, and we're getting the data that we need within a table. We also need to make sure that we've got a hypothesis. So we've got the null hypothesis, and the null hypothesis states that there is no significant difference between the distribution of the 2 species, okay? Or there's no significant difference between the height of a plant in the shade or in the sun, in the other example. So what we're looking for in the null hypothesis is to say there is no association whatsoever. The distribution of the 2 species are random. There's no connection, there's no link, there's no symbiosis, there's no predator-prey, etc., between the 2 species, or the 2 factors, in this case, the height and the availability of sunlight. So the null hypothesis should always start with there is no significant difference between ... The alternative hypothesis is the opposite. So there is a significant difference between the distribution of the 2 species, or there is a significant difference between the height of the plant and the fact that it might be in the shade or in the light. So we've got our data and we've got our hypothesis. And the next thing that we need to do is calculate the so-called expected frequency. I can just see there, pardon for the typo. So to do that, what we need to do is look at the row total, and we need to multiply that by the column total divided by the grand total. So if we look at this one here, the presence, we've got 109, which are present, or 10.9, pardon me, which are present, average 10.9, and the total is over here 21, and the total, if we look at those that are present, is 26. So what we're going to do is the row total, which is 21, we're going to multiply that by the column total, use the column here, column total, and we're going to divide that by the grand total, which is here. So our actual number is 10.9. That's coming from the average of our sample data, and then we calculate the expected using this value multiplied by this value divided by this one. And you can see I've done exactly the same for each one of these cells here. Next thing to do is then to plug that into the chi-squared formula. And the chi-squared is sometimes shown symbolically like this. And what we're going to look at is the sum of the difference between the observed and the expected frequency squared divided by the expected frequency. So sometimes in tables it's quite helpful to put this value here, and then we need to sum this 1, 2, 3, and 4. Okay, and you can see down the bottom that when we've worked that calculation through, we get a value of 7.9. So our calculated chi value is 7.9, and the next job that we need to do is to compare this value against our probability value, which comes from chi-squared probability tables. Before we do that, we need to look at what is called the degrees of freedom, and this will make more sense in just a second. And this is just a mathematical restriction, as you can see here, that allows us to work with a particular significance level as is appropriate. Now, if you really love your maths, okay, you might want to think about this and with your maths teacher work out a proof which tells you why we're doing these things, and you might want to look at the proof of why we use particular degrees of freedom. But the rule for us to use is that we want to have the degrees of freedom, which is looking at the number of rows minus 1, and then the number of columns minus 1. And in the case where we're looking at the distribution of two species, the degrees of freedom will always be 1. So the number of rows